Okay, so good evening, everybody. So today we'll start our lecture for week 11, and the topic for today's lecture is telomerase, and the impact on genomes. So in the previous class, we ended up at telomere, and today's lecture would be all about telomere, the enzyme telomerase, and the impact of telomere length and telomerase on normal functioning of the cell, as well as in the cases of some kinds of diseases. Particularly, we'll be focusing on cancer. So, we'll start with the lecture 1, where we'll study about the importance of telomeres. So, what are telomeres? Telo means end and mere means body. That means something that is present at the end of the body. That means at the end of the chromosomes. So if this is your chromosome, you can see here, this is your chromosomes at the end. The structures which are present, or the sequences which are present at the end of the chromosome, they are known as telomeres. And what kind of sequences are these? These are repetitive sequences. Okay, so telomeres are repetitive sequences that are present at the ends of the chromosome. This structure and this structure, and you can see with the help of microscopy imaging also, you can see that these green colored structures, these are your telomeres. So what is a telomere? It is a 5 to 8 base pair G rich tandem repeats, and it's a repetitive non-coding DNA. That means this DNA does not codes for anything, and these are repetitive sequences which are mostly comprised of guanine. G rich regions are approximately 5 to 8 base pairs. This is what telomere is. Just a minute. Okay. I hope I am audible. Fine. Then you can see, why do we have them? So okay, so we know that, okay, these are so we are talking about we are talking about RNA biology. We are talking about everything related to RNA, and then we talked about transcription and the various kinds of processing that happens. Now we are focusing on telomeres. And one important thing to understand is that these are non-coding DNAs. That means they does not code for anything. Now when they do not code for anything, the question comes, why are they needed, and why are they present at the end of the chromosomes? So they are present at the end of the chromosomes. Why? To protect the chromosomes. Okay, so how do they protect the chromosomes? We shall see in the subsequent lecture, I mean in the subsequent slides, that why telomeres, how telomeres protects the chromosome. They separate one chromosome from another in DNA sequence, and without telomeres, the ends of the chromosome would be repaired, leading to chromosome fusion and massive genomic instability. Okay, so what happens is that, if you see, this is your DNA replication fork. Okay, it looks like a fork. This is known as a DNA replication fork. So one strand is your leading strand, and the other strand is your lagging strand. Okay, leading strand means the synthesis occurs in the 5 prime to 3 prime direction, and lagging strand is the strand which is in the direction of 3 prime to 5 prime. You can see this is your lagging strand. Just a minute, I'll just zoom it. Yeah, you see, this is your lagging strand which is in the direction of 3 prime to 5 prime. Now what happens, that leading strand is in 3 prime to, oh sorry, yeah, this is your leading strand. Now, whatever synthesis has to happen, it always happens in 5 prime to 3 prime direction. This is also something that we know. So, in the leading strand, synthesis is okay. Normally, it can happen. But in the lagging strand, since the directionality is opposite, therefore, these small Okazaki fragments, they helps in the synthesis. Now, what happens is there is a term which is known as end replication problem. What is this end replication problem? That the DNA at the very end of the chromosome cannot be fully copied in each round of replication, resulting in a slow, gradual shortening of the chromosome. What happens that after every replication, the ends of the DNA are not completely copied. So, with each with each replication cycle, your chromosome gradually gets shortened. Okay, your chromosome is gradually decreasing its length with each cycle of DNA replication. So that means after a certain time, after a certain number of replications, your chromosome would be completely exhausted. Chromosome would be completely vanished because of this end replication problem. So to solve this end replication problem, at the end of the chromosomes, we have telomeres. And these telomeres not only protects the chromosome, they also try to keep the length of the chromosome stable up to a certain limit. And also, these telomeres are added with the help of the enzyme, which is known as telomerase. Okay, and these telomerase are the enzymes that adds telomeres at the end of the chromosomes. And telomeres are also thought to be the clock that regulates how many times an individual cell can divide. Telomere sequences shorten each time the DNA replicates. This is important. Why? Because we have just seen that here it is written that after every round of replication, your uh, uh, chromosome gets gradually shortened. That means chromosome is if this is your chromosome, it is getting shortened from the ends. If it is getting shortened from the ends, means telomeres are reducing. So after some n round of replication, after some n round of replication, your chromosome would be like this, very short. So therefore, how many telomeres are there, or the telomeres, they actually tells that how many time a individual cell can divide, because it can only divide up to a certain limit. After that, if all the telomeres are vanished, then it cannot divide, it is completely exhausted. And telomere sequences shorten each time the DNA replicates. Okay, now comes the enzyme which is called as telomerase. So, what is telomerase? So, telomerase is an enzyme that is capable of renewing or adding the telomere or telomeric repeats oblique telomeric repeats onto a chromosome. That means we're talking about that telomeres are added on the end of the chromosomes. Okay, here your telomerase are added, and these are added with the help of the enzyme which is known as telomerase. And one very important feature of this enzyme telomerase is that these are RNA protein hybrid. Okay, this is important. These are RNA protein hybrid which has an internal RNA template and a protein outside. Okay, so structurally telomeres are RNA protein hybrid which has an internally it has an RNA template and it has a protein outside. Then telomerase is nothing but a reverse transcriptase with a single stranded phenotype. That means it adds telomerase only to one of the DNA strands. This is also important. Nothing to explain, self-explanatory. Then important thing is this. Always remember the telomeric repeat. The telomeric repeat sequence is TTAGGG in most of the species. with some slight modifications. Okay, this is the repeat sequence. This is the repeat sequence that is added at the end of the chromosome. Okay. Now, according to a research carried by Dr. Jerry Shea and the colleagues at the University of Texas, it was found that cellular aging can be bypassed or put on hold by the introduction of the catalytic component of the telomerase. Now, we shall see in the subsequent classes that telomeres has a direct relation with aging. So, as long as a cell can divide, it can self-renew itself as long as the telomeres are present. So for example, skin cells. We talk about somatic cells. So uh, cells which are important for, let's say, uh, for suppose for skin. So you see that over time you are gradually, what happens, that skin gets old, it gets shrunken, it gets very fragile. Why? Because many kinds of cells are not replicating that enough, or many kinds of cells are not replicating that can maintain the self renewable or self renewing property of the skin. It is not happening. Many of the skins are unable to do that. That means the telomeres are gradually getting shortened, and because of that, the skin cells are not able to divide. So that is why the age of a cell depends on the telomeres. Always remember, age of the cell is related to telomeres and sorry, telomeres, and in a way it is related to telomerase, because telomerase will only synthesize telomeres. So if you have telomerase externally added into a cell, then this telomerase can synthesize telomeres, and then the aging can be reduced. So that is what this group has shown, that cellular aging can be bypassed or put on hold by introduction of the catalytic component of the telomerase. Now what does it mean? That if this is the whole structure of telomerase, the catalytic portion the catalytic portion is actually the portion which is responsible for uh, addition of the telomeres to the end of the sequence. So if you do not, even if you do not add the whole telomerase enzyme, even if you are adding just the catalytic portion of the telomerase enzyme, even then you can reduce or put on hold the cellular aging process. Okay, fine. Then now we will see in detail what is telomerase. So basically, telomerase is a ribonucleoprotein enzyme complex that has been referred to as cellular immortalizing enzyme. Why it is known as cellular immortalizing enzyme? Cellular, matlab, if you are have attaining immortalization at the cell level, at the cellular level, because cells can replicate or cells can divide only up to the time it has telomeres. As cells will gradually divide, the chromosomes will eventually get shortened, and as they are getting shortened and shortened by each cell replication, there is a limit up to which a cell can divide. But if you give externally, if you provide the cell externally the telomerase enzyme, it can resynthesize the telomeres and the cells can keep on dividing. So that is why telomerase is known as a cellular immortalizing enzyme. So what does it do? So it stabilizes telomere length by adding hexameric, this is TTAGGT repeat, onto the telomeric ends, thus compensating for the erosion of telomeres that occurs in its absence. Okay, this is important. And remember, you can see also here that it's a hexameric repeat. Okay, then telomerase activities observed in fetal tissue. Okay, activities nearly undetectable in somatic cell. Now, it is very important These 2 points are very, very important. Always remember these points. Why I'm saying so? Because even in subsequent lectures also, in subsequent slides also, you will see that this is what decides the activity of telomerase enzyme. Activity of telomerase is not detected in somatic cells. Somatic cells, it is not detected, but it is detected in tumor cells and in germ cells. Okay, so that is why there is an age up to which a somatic cell can divide. But tumor cells are immortalized. They can divide for an indefinite period of time. Also, germ cells also, stem cells also, these are the cells that can divide and they have the telomerase activity that can compensate for the loss of the telomeres. See, whether it is a tumor cell or a germ cell or a somatic cell, after each and every round of cell division, telomeres will be shortened. Telomeres will get shortened irrespective of the type of cell. Telomeres will get shortened in a somatic cell also, in a germ cell also, in a tumor cells also. But these cells are growing indefinitely because they have the enzyme telomerase. That means these cells have an enzyme that can replace the eroded telomeres from the chromosome. Is it clear what I'm trying to tell? Are you understanding? Are you following? Just say yes or no. Okay, fine. Now, this is about what is the function of telomerase. Now comes how that function is attained or is done by telomerase enzyme. So, research also shows that counter that controls the wasting away of the telomere can be turned on or turned off. The control button appears to be on telomerase, which can rejuvenate the telomere and allow the cell to divide endlessly. Most of the body contain telomerase, but it is in the off position, so that cell is mortal and eventually dies. So, if I say telomerase, if here they have written telomerase in on and off position, what is the meaning of telomerase in on and off position? Here it is saying that in most of the body, telomerase is present, but it is in off position. That means telomerase is present in somatic cells also, but it is in a off position. So what is off position describes here? Can anybody tell me? What is the meaning of off here? If a gene is present and it is off, that means what?
Not expressed. Not expressed.
Uh, Kavita, your voice is not coming. I don't
It
know
is
whether
not
it's my problem or.
expressed. It is not
Hello? Yes? Uh,
expressed.
Unable to hear your voice. Suppressed state. Okay, Okay, so Madhavi has said suppressed states. Kavita, I'm unable to hear your voice. I don't know whether the problem is from my end or your end. You can write not expressed. Yes, exactly. You're saying it completely right. That off means that the expression is not there. It is not expressed. So we have already, I don't know whether I have told you this earlier or not, but just for your Kavita, I'm unable to hear you. Sorry. So I think there is, I don't know whether the problem is from my end or it's the some connectivity problem, but whatever you want to say, you can just type it. I hope my voice is coming to you. Am I audible to everybody? Okay, fine. So, okay. So one thing that I have told you in some previous lectures, that all the genes are present in all the cells. It depends on the expression. So the expression of genes is cell specific and context specific also. So genes which are responsible for eye color, they are present in lungs also, but they are not expressed in lungs. Likewise, telomerase is present in somatic cells also, but it is not expressed in somatic cells. So it's not the presence of a gene that is important, it is the expression of the gene that plays a key role. Okay, now we'll see further. So how does this all link together in the laboratory? Cells in tissue culture with introduced telomeres have extended length of their telomeres. They have already divided for 250 generations past the time they normally would stop dividing and are continuing to divide normally, thus giving rise to normal cells with the normal number of chromosomes. What does it mean? It means that if you are externally supplying telomerase enzyme to the cells, then what happens? That they keeps on dividing and dividing, and they have an extended length of chromosomes, which shows that telomerase is indeed the enzyme which is responsible for adding telomerase at the end of the chromosomes. And telomerase is the enzyme which adds telomeres at the end of the chromosome. And telomeres decides how many times, how many number of times a cell can divide. Okay, so two things are there, do not get confused. One is telomerase and another one is telomere. Okay, one is telomerase and other is telomere. Do not get confused. Telomerase is the enzyme that adds telomere. Okay. Always remember, if you have, you might already know this, but I'm just telling you that all the enzymes have A's in them. A's. For example, ligase, polymerase, phosphatase, nuclease, and any other enzyme for that matter. Peptidase, carboxypeptidase. These are the enzymes which are responsible in digestive system. Proteinase, DNase, RNase. So all the enzymes have A's in them. So even if you get confused between telomerase and telomere, the one which has A's is always the enzyme. Okay. Now we'll just see two questions about this lecture one, and then we'll move further. So identify the correct telomere repeat sequence, and here I am talking about in humans. So these are your four options. I'm giving you 1 minute to think, and then you give me the answer. Okay, I'm switching the screen. So Kavita has answered A. Okay. Any other? Nobody wants to give a try? Think. At least I want one more answer from anybody. Nobody? Okay. Fine. See, TTAGGG. How to remember it? Hmm. So do you think there is any other correct option in this? I don't think so. The question that they have provided, there is no correct option. Just a minute. TTA triple G. So is there any TTA double G is there? TAA the TTAG. So there is no correct option actually. You see, there is no correct option here. Yeah. So the correct answer is TTAGGG. How to remember it? T2AG3. T2AG3. Like this, you can remember because there are two T's and three G's. T2AG3. Always remember this is a 1, 2, 3, 4, 5, 6. It's a hexameric sequence. Hexameric sequence, that means there are six nucleotides are there. 2T, 1A and 3G. T2A, G3. T2A, G3 or T square A G cube or T2A, G3. Like this you can remember. Okay. Now next question is, telomerase is a RNA protein hybrid that has an internal RNA template and a protein outside. Is it true or false? We have discussed this just now. Think. Okay, I'm switching the screen. Hmm. Kavita T. Okay, true. Yes, this is a true. We have discussed that telomerase is a RNA protein hybrid that has an internal RNA template and a protein outside. For those who have missed, I'll just repeat it. See, hmm. It's an RNA protein hybrid that has an internal RNA template and a protein outside. Okay. Now we'll go to quickly lecture number 2. So in lecture number 2, it is talking about telomerase in aging. So up till now, we have talked that telomeres decides that how many times a cell will divide, and how many times a cell will divide tells that how much the cell has the property of self renew. Self renewing means how many times the cell is able to divide itself and make a new copy. That means in a way cell division is related to self renewal capacity of the cell. That means if you have more telomerase in a cell, aging would be what? What would be the effect on aging? Aging will come early or it will be delayed. If more telomerase is there in a cell, will aging be delayed or aging will come fast? Tell me. If there is more telomerase in a cell, aging would be delayed. Yes. So aging would be delayed. Why? Because telomerase will add telomeres, and as long as you have telomeres keeps on getting added, your cell will keep on dividing. And if your cell is if the cell keeps on dividing, that means it is able to self renew itself, and therefore the aging would be delayed. Okay. So that is why this telomerase and aging are a very widely learned concept today. People do a lot of research on telomerase and aging and how telomerase can affect aging, and a lot of research is going on telomeres and telomerase. It's a very hot topic for today's research. So how does now we'll see how does telomerase work. So telomerase work by adding back telomeric DNA to the ends of the chromosomes, okay, thus compensating for the loss of telomeres that normally occur as cell divide. Okay, so most normal cells do not have this enzyme, as thus they lose telomeres with each division. That means normally somatic cells. That is why each cell has a lifespan. No somatic cell is immortal. Each somatic cell has a lifespan. That means up to a certain limit only it will divide. Up till a certain number only it will divide. After that, that cell will die. And what decides the number of cell divisions? Telomeres. If the telomeres are present, then only it is able to divide. Otherwise, it will get exhausted and it will die. So mostly cells do not have this enzyme, or the enzyme is not expressed, and they lose telomeres with each division. In humans, telomerase is active in germ cells, in vitro mortalized cells, and the vast majority of cancer cells, and possibly in some stem cells. This is important point. Okay, remember that where all this telomerase is active in humans. Okay, you can see here that this telomerase is active. This telomerase enzyme is active in germ cells, in vitro mortalized cell means in vitro cultured tumor cells, the vast majority of cancer cells, and in some cancer stem cells also. And high telomerase activity exists in germ cells, stem cells, epidermal skin cells. Now, epidermal skin cells. Why you think that epidermal skin cells and follicular hair cells have high telomerase activity? Can anybody tell me? Why epidermal skin cells and follicular hair cells, these two cells, okay, why do they have a high telomerase activity? Anybody? Just think randomly. Fast dividing, yes. So if you have a cut, just a minute. Yeah. So if you have a cut or a wound somewhere, and after sometimes you will see that the wound is healed. That means there's a new formation of cells. Or if there's a scratch, it is healed. It is because of telomeres. Or your hairs. You see, whenever you comb your hairs, I mostly see all the ladies' hair, and we all have a general problem that our hairs are falling like anything. And we are so conscious that one day we all will go bald because our hairs are falling like anything. But what is the thing that although with every wash, with every comb that we do, so many hairs comes out, but there is production of new hairs, and you have new hairs on your head, because these are self renewing itself, and that self renewal property is coming because of telomerase. Okay, so telomerase are actually important in your skin cells and your hair cells to maintain it, to replenish the ones that have died, to replenish the cells that have died. And this high telomerase is coming because telomerase enzyme is active in these cells, in these areas, in germ cells, in stem cells, in epidermal skin cells, in follicular hair cells, and cancer cells. Okay, so some cells are immortal because their telomerase is switched on. So cells which are immortal means that never dies. They keeps on dividing and dividing and dividing because telomerase is continuously switched on. And because telomerase is continuously switched on, what happens? That it keeps on adding back the telomeres and the cells never dies. Examples of immortal cells are blood cells and cancer cells. Cancer cells do not age because they produce telomerase, which keeps the telomere intact. Okay. Now, aging, we're talking about life and death, how aging is related. So as soon as your cells are dividing, with each division cells are getting short, and this with each cell division, with each shortening of DNA, cells are aging. So an aged cell will have very few telomeres or no telomeres and telomerase, whereas a young cell at the starting, it will have a lot of telomerase. Okay, now, so genetically, on the genetic level, if you see, then these telomeres, they control gene that has been mapped to. This is just the location of the gene that it controls, and there's nothing to explain. You can just read it. So this is important that healthy human cells are mortal because they can divide only a finite number of times, growing older each time they divide. Thus, cells in elderly person are much older than cells in infant. So, you can see, that elderly persons in infant or in youth, you have a very glowing skin. There is a brightness on your face. And as we get old, as our age increases, you know, we can see visible difference on our face. The morphology remains the same, but you can see that now you have a little bit more wrinkles are there. Skin is also becoming a little loose. I mean, that freshness that a child has or a youth you have, that won't be there as you start aging. Why? Because now cells are getting old. As you are getting old, similarly your cells are also getting old. And in molecular biology terms, getting old means that telomeres are decreasing. Okay. So with every cell division, telomeres are decreasing, and because telomeres are decreasing with every cell division, the cell is getting old. Okay. And ultimately, up to a certain point, the cells will die. So that is why the to maintain the self renewable or to maintain that glow in aged people, because cells are dying now, so therefore that self renewal capacity is lost. And that is why if you have a cut or a wound, it's not very easy to heal it. And even in your face, the cells that can just tighten up your skin, they're also not very much present now. So this is how telomerase and aging are related to each other. The discovery and function of telomeres and telomerase is the single most important discovery in the field of anti-aging medicines. Okay, so what do you think the anti-aging medicines can have in them? Can anybody tell me what component, if suppose if I want to make one anti-aging medicine, I know nothing about it. What do you think should be the main ingredient in that anti-aging medicine? Anti-aging creams. There are so many anti-aging creams available in the market now. Tell me. You can type. Anybody? Hmm. Okay. So, Kavita has said telomerase and Tarkeshwari has said collagen. Yes. Collagen is also very important. Um, Here we are talking more about at a cellular level basis. So, cellular level basis for us, for suppose for skin, how skin can maintain its glow, how we can reduce the aging if the cells they keep on rejuvenating themselves, if the cells keep on renewing themselves, that means the cells should keep on dividing. And to keep the cells dividing, we need the component that is responsible for division, and the component that is responsible for division is telomeres, because as long as you have telomeres, the cells will divide. And what adds telomeres in your cells? Telomerase. That means you need to have telomerase enzyme so that telomerase can keep on adding the telomeres on the cell, on the chromosomes, and the cells can keep on dividing. So the division of the cell is very important. That is why, yeah, I don't want to go to that much detail. That this is enough for you as of now. Okay, now we understood that okay, telomerase is there, and how telomeres and telomerase is related to aging and senescence. Now comes that what causes aging. We say that aging happens, so what causes this? Because cell division is a finite biological function and is inexorably tied to the aging process. That means cells are immotile or motile. Tell me, somatic cells are immotile or they are motile? as in
Mortal. Mortal.
motile. That means they will keep on dividing only up to a certain limit. After that, they will die. Okay, so that cell division is a finite biological process, and this is defined by a number which is known as Hayflick number. And this Hayflick number is important. Hayflick number describes the number of times a cell can divide. Important. Okay, marking it. So, how many times a cell can divide? It is described by a number, and that number is known as Hayflick number. Hay, Hayfli. It's better written there only. Flick. Hayflick number. So Hayflick was the name of the scientist who was the first to suggest the relationship between cell division and mortality, but the mechanism was not initially understood. Okay, so now we'll see in detail about the end replication problem that we talked about at the start of the class. So DNA polymerase can only synthesize DNA in the 5' to 3' direction. Okay. So there are two strands just we talked about. I'll just read it first and then again go back to that image. Okay. Consequently, DNA synthesis does not extend to the very end of the lagging strand. So there are two strands. If you know the basics of DNA replication, so DNA has two strands. One strand is the leading strand, another strand is the lagging strand. So in the lagging strand, you have Okazaki fragments, and synthesis occurs in Okazaki fragments. So the name itself suggests that leading strand because the replication occurs first, and lagging strand because it lags in the replication. That is why it is known as lagging strand, because it is lacking. That means in the two strand, the leading strand replicates first and then your lagging strand replicates. So what happens that if this is your lagging strand, so what happens that DNA synthesis cannot occurs at the end of the lagging strand. So the ends of the lagging strand with each round of replication gets shortened, and this is known as end replication problem. That means the end region of the lagging strand is not replicated, and with each replication cycle the ends of the lagging strands gets shortened and shortened. Over a period of time it is completely vanished, and then your cell dies. Is it clear? So, in the absence of a protective mechanism, the end replication problem means vital genetic material would be lost during each cell division. Okay, and that is the reason why germ cells have telomerase enzyme. Because germ cells are, why you think that germ cells are more important than somatic cells? That germ cells have telomerase and somatic cells do not have telomerase. Why you think germ cells are more important? Tell me.
It produces gametes.
Can you just increase your voice, please?
It produces gametes, ma.
Okay,
Gametes.
so okay, fine. Yeah. So germ cells are more important because they produces gametes and they are responsible for the production of an offspring. So in an offspring, you cannot have that because of end replication problem, genes that codes for body color or that codes for some vital organs or that codes for certain proteins that are very responsible for your digestion are lost. Then what will happen? The offspring that is produced, will it be a normal? No, it will not be a normal offspring. There will be so many errors in the production in the embryogenesis that the offspring that is produced would not be completely fit and fine. Therefore germ cells, you cannot afford to lose a portion of germ cells with each round of cell division. That is why germ cells has a high expression of telomerase enzyme. Somatic cells can afford because these are responsible for the self renewal capacity. So even if a cell dies after some times, other cell will come and take over it. So no problem. Okay. So Now we talked about the end replication problem. So what is the end replication problem? That the end portions of the lagging strand of the DNA, it is not completely replicated. And therefore, with each round of cell division, the chromosomes get shortened and shortened and shortened. Which one? The lagging strand of the DNA. So this end replication problem is solved by the enzyme telomerase. So telomerase solves the end replication problem by protecting the cell from the loss of critical coding base pairs. And this protection is specifically needed in germ cells and in stem cells. Okay. Is it clear up till now? Is it clear to everybody? Up till now, is it clear? Okay. Now we'll just try to solve one or two questions. Yeah. DNA end replication problem is alleviated by RNA polymerase, reverse transcriptase, telomerase, telomeres, and telomerase. Okay. Think 1 minute and then I'll switch the screen. Think. Okay. So Madhvi has said D, Kanchana has said D, Janara has said D, and Momita has said C. Let me see. Yeah, yeah, yeah. The correct answer is D. Why correct answer is D and not C? Because telomeres can be added with the help of telomerase. So telomeres are already present. Telomeres are already there in the uh, chromosomes. So it should be telomerase. Am I correct or wrong? Tell me. I just want to see what you think. It should be telomeres or telomerase. These two are very confusing. Okay. Each division they get shorter. So telomerase is correct to add more telomeres. Yes. So basically, what we are saying, what is the meaning of alleviated? So alleviated means something which can be reduced or which can be made less potent or less severe. So how we can correct or rectify the problem of end replication? So we can rectify the problem of end replication by adding more telomeres, which can be added by the enzyme telomerase. So now this question is given in your assignments, and they have told us that the correct answer is telomeres, that is C. But I think the correct answer should be telomerase, because ultimately telomerase is the enzyme that is adding telomeres. But this question is very context dependent. Suppose if you were given only these three options: RNA polymerase, reverse transcriptase, and telomeres, at that point you can choose telomeres, not telomerase. Okay. But when both are given, according to me, I think telomerase should be, looks like a better option. But I don't know why they have written telomeres. But if you just try to think about it, telomeres are already there. Every chromosome has telomeres. The problem is that they are getting shortened and shortened with each replication cycle. That means the correct solution would be to give telomerase enzyme, so that it can renew the telomeres which are getting shortened with each replication cycle. Okay. Fine. Next question is, telomerase come under the category of nuclease, reverse transcriptase, DNA polymerase, or DNA ligase? Think. Think, and I'll switch the screen 1 minute.
Ma'am, previous question, it is telomere or telomerase, ma'am, the correct
Uh,
answer?
sorry. Can you just type what you want to say? I can hear that you're saying, but it's not very clear. Um, your voice is not very clear. It's very low. I don't know why. Mm, yes, can you repeat now?
Previous question, is it telomere or telomerase? The
Yeah.
answer is
So the answer is telomerase, because the problem can be reduced if you have telomerase enzyme that can add more telomeres. Okay,
okay.
but I think but in your this thing, I mean, whatever the answers that they have given to us, they have given to us as telomeres. But I don't think
Okay.
that telomeres should be the correct answer, because you also think no, every chromosome, it already
Uh,
has telomeres, right?
okay.
So,
Okay.
but the problem can be reduced by the addition of telomeres, and that addition can be done only by telomerase. And that is why in most of the anti-aging drugs or whatever, in genetic engineering or whatever they are doing new research, they are focusing on telomerase enzyme and not just telomeres. Because if you're supplying telomerase enzyme, you're solving the problem for the indefinite number of time. But if you're just adding telomeres, you're solving the problem up to a certain number of replication. After that, again telomeres are vanished. Then what you will do?
Okay.
That's why.
Okay.
Okay, thank you. So yeah,
Okay.
so this is the question. Next question is, telomerase come under the enzyme category: nuclease, reverse transcriptase, DNA polymerase, and DNA ligase. So I think people have answered it. Okay, B, reverse transcriptase, yes. So telomerase comes under the category of reverse transcriptase, correct. Okay, so now we'll come to the next, yeah, lecture 3. So this week, mostly all the slides are focusing mostly on telomeres and the aging and a little bit about cancer. So the topics might be a little repetitive. It might look a little repetitive, but then again, as many times as we'll read about it, it will make our concepts only clear. Okay, so telomere length as marker for aging. Okay, telomere length as marker for aging. So if telomere length is high, then what will happen? If telomere length is, I'm sorry for my extremely bad handwriting. I'm not very used to writing on this writing pad. That's why, even though I'm writing on this pad for the last 1 year, still I'm not very used to it. Anyways, so tell me, if the length of the telomere is high, what would be the effect on aging? Anti-aging, yeah. So if telomeres, length of the telomere is very high, so aging would be prolonged. I mean, aging would come after a very long time, or it will come early. Aging will come early, or aging will come in a long time, or in a greater span of time. Okay, fine. And if telomere length is less, what will happen? Aging will come early or not? Think and tell me. Early aging. Okay, fine. So that means if telomere length is very high, high length of the telomere means that it can replicate for a greater number of time, for it can have more number of replication cycles. More number of replication cycles means aging will come after a long time. But if telomere length is very short, what will happen? Less number of replication cycles, aging will come early. So I think now this concept is clear to everybody that there is an age dependent attrition of telomere length which loses ranging from between 30 to 150 nucleotide pair per replication depending on cell type. Means after every cell replication, approximately 30 to 150 nucleotide pair of telomeres are lost after every replication. Cell division or telomere shortening continues until a critical telomere length is reached, at which point cell is forced into senescence and can no longer replicate. Cellular senescence prevents replication of incomplete or damaged DNA. It is very important and we shall see in it subsequent slides also. I am just telling you, this is suppose the length of your chromosome and I am marking this as some critical point. With each replication cycle, your so I cannot do what I think what I'm thinking. Fine. So with each replication cycle, in first cycle, this much removed. Second cycle, this much removed. Third cycle, this much removed. Fourth cycle, this much removed. Fifth cycle, this much removed. Sixth cycle, this much removed. Seventh cycle, this much removed. Eighth cycle, this much removed. So even after 8 cell divisions and the removal of this much cycles, cell was able to function normally. Means it has a threshold. Like everybody has a capacity up to which we can um, take things, and after that we get saturated, and then there is an outburst we cannot take after that. So just think of this green line as the limit of the cells, that okay, I can tolerate up till this much divisions. After this, I cannot tolerate. After this, I will become damaged. So if you want to reduce my length, you can reduce my length up till 8 divisions. After 8 divisions, I am not no longer a normal cell. So just let me die. Otherwise, I will become damaged and I will cause problems in the cells, in the body. So what happens? Here I am just for your convenience and just as an example, I'm telling you 8 divisions. So up till 8 division, this cell X has replicated. After 8 division, it has reached a limit that if it replicates further, it will give a damaged product. So it's better that this cell should die. So that is what is written over here, that cell division or telomere shortening continues until a critical telomere length is reached. This is the critical telomere length, okay? Until a critical telomere length is reached, at which point the cell is forced into senescence and can no longer replicate. Why? Because if it keeps on replicating, then it will replicate incomplete or a damaged DNA. So it's better that to replicate an incomplete or damaged DNA, it's better that the cell should die. Okay. So this is what telomere length as a mark of aging will tell us. Then telomere mediation of gene expression may explain the relationship between telomere length and aging. This we have already just solved the problem, how telomere length is related to aging. Okay. So now what happens that you have in DNA, you have a primary structure, you have a secondary structure, you have a tertiary structure, and you have a quaternary structures. So what they are saying is that telomeres affects all the 4 structural levels. That means these, I am talking about DNA. So DNA has a primary structure, which is the structure of the sequences. I mean the sequence of the ATGC, secondary structure, then tertiary, and then quaternary structures. So primary and secondary structure means telomeres protect the integrity of the base pairs and preserve basic genetic information. That means telomeres are so important for a cell. We're talking about the importance of telomeres. Telomeres are so important. They protect your chromosomes. Okay. And they are not only required at one level, they are not on they do not only protect your chromosomes at primary structure level. At secondary structure level also they are protecting. At tertiary and quaternary structure level also they are protecting. So how they are protecting at the primary and secondary structure level? They protects the integrity of the base pairs and preserve the basic genetic information. Because ultimately, see, the most important, that is why we say the most important part of primary, secondary, tertiary, quaternary structure is the sequence of the base pairs, ATGC sequence. Because if this ATGC sequence is disturbed, then everything which comes downwards of it will get affected. That is why even in our education we say that clear your basic clearing is very important. Your concept, your basics of the concept should be clear, your logic should be clear, because these are the building blocks that is going to decide the whole building that you're going to make of with your knowledge. Similar, likewise it comes at DNA level also. At the primary structure, whether we are talking about a protein or a nucleic acid, primary structure is the most important part, because it is the building block which is going to decide your primary structure will decide your secondary, tertiary and quaternary structure. So telomeres, it protects the primary and secondary structure by protecting the integrity of the base pairs and preserving the basic genetic information. Whatever the sequence of the base pair, it protects. Then, how it affects the tertiary and the quaternary structures? As telomeres shorten, they may exert different steric effects, resulting in changes in the shape of DNA. Yes, and these changes may in turn affect the genes exposed and available for transcription. That means if secondary structure of the DNA was something like this, and this structure has put this promoter and this enhancer in very close proximity, therefore the transcription of a gene X is happening, and this is because of the telomeres which are present. If telomeres are not there, then what can happen? That the structure can be somewhat like this. Now this is the promoter and somewhere this is the enhancer. Here they were so close in proximity because they have looped out. So telomeres can change the shape of the DNA, and the shape of the DNA can change the phase of the transcription, because now it can know the promoter and enhancer cannot interact with each other, and not just promoter enhancer interactions. Suppose if your structure was early very loosened up, so there's a space for transcription factors to come and replicate. But now, because of the reduction in the number of telomeres, your structure has become so compact that there is no space for the transcription factors to come and bind and do the transcription process. So, in any ways, in any matter, in any form, your telomeres help only. So, at all the four structure levels, primary, secondary, tertiary, quaternary, it is protective. So it's more like a genetic clock. It's a protective cap. Why? See, this is your chromosome. These are the ends. And these are protected because of telomeres. Now, because telomeres are present, so it cannot be acted upon by exonucleases. Nuclease cannot come from here and it starts digesting it. So it's like a cap. Just like a cap prevents our face from the direct light and the heat. Similarly, this telomere cap is protecting the chromosome from the direct action of exonuclease. Is it clear to everybody what I'm trying to tell? Are you people following? Following me? Just tell me or give a thumbs up or whatever. Okay, fine. Hmm. So, just what I said, that it protects the chromosome from recombination, exonuclease degradation, end to end fusion. It distinguishes telomeres from DNA double-stranded breaks and it prevents oncogenesis. So these are the functions of telomeres. Now, what is the cap? So we talk about that in the end, it is a kind of a cap, and this is nothing but a nucleoprotein complex. We already talked about the number of different proteins that can bind to telomeres. What are the different proteins that can bind to telomeres? Single stranded DNA and double stranded coat and protect the telomeres. This is nothing. This is just to explain you that this cap, what all functions it does. So this telomere cap, it protects the ends. It helps in the protecting the integrity of the chromosome, and it protects, and it also helps as a surface. It also provides a surface for other proteins to come and bind to it. Okay, and this I have just written, you can just go through it. This is nothing but just the importance and what are the functions of telomeric proteins. Nothing to explain, this is just self-explanatory, just simple sentences. Okay, now we'll move further. Okay, we'll solve some questions. Fine. So telomere is a repetitive sequence present anywhere on DNA. True or false? Think. I'm giving you 1 minute to think and then I will switch the screen. Think. Okay. Hmm. Janara has said yes, true. But then she said false. Okay, Tarkeshwari has said true. The answer is okay. So people who have said true, can you explain me why you have said true? What was the reason? Why you think it is true and not false? Give me one explanation, please. Anybody? Anybody means okay. Madhvi, you said true. Can you explain why you said true? Why you think it is true? Hmm. Yeah. Okay. I'm sorry if you are hearing some um, chewing noises because I haven't had my lunch and I didn't got any time, so I'm just eating some snacks in between because I haven't had anything since morning. Anyways, so the answer is false. Why? It is a repetitive sequence, okay. But is it present anywhere in the DNA? No. It is present at the ends of the DNA. Telomeres are specifically present at the ends of the DNA. They are not present in between or somewhere here or somewhere here. No. They're only present at the ends of the DNA. That is why telomere is a repetitive sequence present anywhere of the DNA. It is a false statement. I have told you in the very first slide, if you remember, you see, these are the telomeres which are present at the ends of the DNA. You see here also in this image also you can see this green color portion. This green colors, these are your telomeres. And you can see this is a published paper. In nowhere you can see telomeres in the middle or any other position except the ends. Okay. So the answer would be false. Next question is, telomere length is maintained constant in all chromosomes. True or false? 1 minute and then I switch the screen. Okay, I'm switching the screen now. Yeah. Okay, Madhvi. So Janara has said false. Okay. Anybody else who wants to give a try for this question? Telomere length is maintained constant in all chromosomes. What should be your answer? Nobody wants to give it a try. Kanchana, Lakshmi Kanta, what do you think? It should be true or false? Madhavi, what do you think? It should be true or false? Just any guess. This it's not a exam or test. We're just having a friendly discussion here, trying to learn from each other. Okay, fine. So I always want my class to be very interactive. And why I say so? Because I'm not a teacher. I'm just a student. I'm also a student. I'm a PhD student, and I'm just trying to discuss and share my whatever experiences I have. And I'm sure that you will have even more experiences in knowledge and experimental wise also. We're just exchanging knowledge and ideas here. Okay, so Madhvi has said maintained, don't know, but they have finite number of divisions. Okay, okay, fine. I understood what you're trying to say. So the answer is false. Why? Because see, telomere length is maintained constant in all chromosomes. So I'll just tell me, uh, if I have a germ cell. Okay, I'll just do it in a next. I have a germ cell and a somatic cell and a cancer cell. These are three different types of cells. My question was that telomere length is maintained constant in all chromosomes. So, what will happen to the telomere length in somatic cell, germ cell, and cancer cell? Now, tell me, in germ cells, will telomere length be maintained, or they will reduce after certain number of times, after certain number of divisions? Will telomere length will get reduced, or it will be maintained in germ cells? It will be in germ cells, it will be maintained. That means the length of the telomere is continuously getting maintained here in germ cells. Here it is maintained. In cancer cells also it is maintained because it is immotile. But in somatic cells, it keeps on getting reduced. With each cell division, the length of the telomeres gets reduced. Whereas in the case of germ cells and cancer cells, with each cell division, the length of the telomere is maintained because telomerase is active here, telomerase is active here, but telomerase is not active here. So the question was, telomere length is maintained constant in all chromosomes? The answer would be no, because in somatic cells, the length of the chromosome, the length of the telomeres keeps on getting reduced with each cell division. Is it clear to everybody what I'm trying to tell?
Yes, ma'am.
Okay. So it is not maintained constant, but it is maintained constant in germ cells, in cancer cells, and in few types of stem cells. Okay. Now we come to the next topic, lecture number 4. Now this is important and very interesting. So this telomeres topic is actually very interesting. You see, for so long we were studying really monotonous kinds of topics, but this is a break and very refreshing topic. Genetic clock. Now let's see. See here, very interesting. This is a graph. This is telomere length and this is cell replication. So in germ cells, these are germ cells. You can see with each round of cell replication, the length of the telomere is approximately the same. It is constant. It is not going down. That means the telomere length is maintained constant in germ cells with each replication. Because, and you can see telomerase plus, that means telomerase is present here. Stem cells also, it is maintained constant. Reduction would be there, but very less. You can see approximately it is maintained constant only because telomerase is present. But in somatic cells, telomerase is not present. So you can see there's a steep decline in the telomere length with each round of cell replication or cell division. You can see earlier it started from here. It is going down, 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 down. Now at this position, telomere is getting activated. And if some modification happens, that somatic cell can turn into a tumor cell. And when a somatic cell turns into a tumor cell, telomerase is there and it becomes immotile. It is a cancerous cell. Means a normal somatic cell, a normal somatic cell has turned into a cancerous cell. Some mutations, some modifications has happened over here, which has turned a normal somatic cell into a cancerous cell. Okay. Or if the telomerase activation does not happen, then it undergoes the senescence, means it dies. This is the genetic clock. And from this also you can see that the length of the telomere is not constant across all chromosomes. It changes. It keeps on changing. Okay. So what is written over here? That telomere length was accessed in 143 normal unrelated men and women, and individuals with shortest telomeres had significantly decreased survival rates and higher mortality due to heart diseases, due to infectious diseases and all that. That means if your telomeres are short, your survival rate is also very less. So lesser the length of the telomeres, lesser is your survival rate. Is this graph clear to everybody? Is this graph clear? Just tell me yes or no. Okay, fine. Now, so today's class is very light. Things are very light, and all you need to understand is the logic behind it. It's very interesting also. Now, again, you see relationship between telomere length and age related conditions. Okay, these are different age related conditions. um, Hypertension, metabolic syndrome, diabetes, and I don't know what this MMSC is, but okay, these are some kinds of diseases only. You can see these are the ones with diseases and these are the ones without diseases. And this is T by S ratio. This T by S ratio means smaller the T by S ratio, if this T by S ratio is less, your chance of getting a disease with age is high. That means this T by S ratio, it should be high only. Then only you are healthy. You can see in each and every situation, without disease, your T by S ratio is so high. You have a great number of telomeres. The length of the telomere is very large, very huge. Okay. Here also you can see cardiovascular diseases. Here you can see with no disease and with disease. So with no disease, you can see it has such a high T by S ratio. Whereas with disease, they have such a low T by S ratio. So measured mean telomere repeat copy number to single gene copy number is T by S ratio. Means per gene, how many telomeres you have. Per gene, I'm just trying to explain you. It's very easy. Just a minute. Give me 2 minutes. Yeah, I'm just telling you T by S ratio. This is a gene X, and this is a gene Y. This has 6 telomere repeats. It has 3 telomeres repeats. So it would be T by S ratio would be 6 by 1 or 3 by 1. Why? Because it is mean telomere repeat copy number to single gene copy number. So which has a higher telomere repeat number? This one. So if your T by S ratio is high, then the chances of disease is low. If T by S ratio is low, chances of disease is high. So T by S ratio is inversely proportional to disease occurrence. Is it clear to everybody what I'm trying to tell you over here? That per gene, how many telomere repeats you have. So if you have more telomere repeats, numerator would be great. I mean, numerator would be more. And then what will happen? Your T by S ratio would be more. If you have less number of telomere repeats per gene, numerator would be less and T by S ratio would be less. Okay. So if you're, so just, you just need to understand that T by S ratio, it should always be high to maintain a healthy condition. You can see these all are the samples without diseases. Without disease, your T by S ratio is so high. Here also same thing. Okay. Now let us see over here. Hayflick limit. We were talking about Hayflick limit. No. See, same diagram that we have discussed. This is the telomere length and this is the cell division. You can see with each cell division, even with each cell division, germ cells are maintaining constant telomere length. Whereas somatic cells, you can see somatic cell, there is exponential decrease in the telomere length. Okay. And this limit is known as a Hayflick limit. Why? Because at this limit, at M1, senescence occur. So Hayflick limit is a situation where the cell has to take the help of P53 and retinoblastoma proteins to make a living. Okay. So Hayflick limit is the limit where the cell if it is taking the help of P53 anything, just remember that Hayflick limit describes the limit up till which a cell can replicate without getting damaged. That means a limit cell can, just a minute. I'll just explain it. Hayflick limit is up to which a cell can replicate without any damage, and mostly cells should die after Hayflick limit. Okay, so here you see this is the Hayflick limit. So after Hayflick limit, the cell should die, senescence should come. But if a cell crosses this Hayflick limit and keeps on replicating, then this portion leads to genetic instability, because the cell is saying, the chromosome is saying that I am not good enough to carry out the normal functioning. Please let me die. Please, the chromosome is saying to the cell that please let us die, because I am no more a functional chromosome. I do not carry all the informations and I do not carry the information property. It is better for the body to let us die. But the cell is saying no, I will keep on replicating and I don't care what is happening to the cell. So what happens after the Hayflick limit? If the cell is getting died, if the cell is getting senescence, then it is completely fine. But if the cell keeps on replicating, then crisis comes. So M1 refers to senescence and M2 refers to crisis. So, Hayflick limit is reached at a stage at M1 senescence, that is the first initiation phase of senescence. Division continues with lots of difficulty and it reaches to M2, and it is called a crisis phase. Crisis phase, you end up having a genetic, I mean, in crisis phase, there is genetic instability. There is genetic instability in crisis phase. So this genetic clock is nothing but a interrelationship between the length of the telomere and the number of times a cells can divide further. Okay. So is this concept of genetic clock clear to everybody? Yes or no? Just tell me. Is this concept of genetic clock clear to everybody? Shall I proceed further? Okay. Fine. Now, this is just for your reading, that what are the situations in which cancer is progressed. So it gets progressed when it bypass the cell cycle checkpoints, when the tumor suppressor genes are not functioning. It continues to divide very short telomeres that we have just discussed, no longer protects the ends. Cells enter secondary proliferative block called crisis, characterized by short telomeres, end-to-end fusion, anaphase bridges, and apoptosis, show rampant genomic instability and widespread cell death. These are the situations where situations, or you can say rather characteristics of a cancerous progression. Okay, now let us solve these two questions. Shortening of the telomeres is also observed in the somatic cells and stem cells. True or false? Just read the sentence carefully and then try to answer. I will give you 1 minute for this question. Try to think. Okay, let me just switch the screen to see the answers. Right. Yeah. So, uh, uh, yeah, sorry, I think there was some technical issue. I'll just immediately share the screen. So I'm sorry because I'm again, there was some technical issue. So can you just write your answers again, what you have written? Yes or no, why, or true or false? Our question was this. Shortening of the telomeres is also observed in somatic cell and stem cells. Can you just rewrite your answers here? Are you able to hear me? Can you hear me? Yeah, okay. So Janara has answered true. Kavita has answered true. Okay, fine. And I will say false. Now tell me why I am saying false. Is this statement completely correct or partially incorrect? Tell me. That's why I was saying that try to see the statement very clearly. See, is there some incorrect thing here? Is it completely correct? Hmm. Partially incorrect. What is the incorrect thing over here? Incorrect statement over here.
Stem cells.
Stem cells. Yes. So the answer would be false. So that's why I have made this question. I myself made this question that shortening of the telomere is observed in stomatic cells, but it is not observed in stem cells. That means this is a false statement. It is all observed in somatic cells only. This should be the question. Why I have given this question? Because when you solve your CSIR questions in your C section, you have given options. You are given options like this only. It is observed in somatic cells and stem cells. It is observed only in somatic cells. Both A and B are correct. Like this, you have questions. So you should always try to see and understand the question very carefully. Okay, you know the concept, you know the answers, you know everything, but you just quickly go through the sentence. You see how somatic cells are okay, somatic cells, it's true. But no, here stem cells is also written, so it becomes false. Okay, next question is smaller T by S ratio can be a consequence of age, stress, more intake of. Okay, this question I think we will deal after we study this lecture. After this lecture, I will I'll just put this question here. Okay, after at the end we'll solve this question because these topics we haven't studied up till. Fine. Now we come to lecture 6, which is the last lecture of today. So this is what we have been studying for such a long time. Again, just to revise, because this is how the syllabus is framed. So the telomere length, as it comes down, it will reach a Hayflick limit. Yes, that is the limit in which the cells can no longer divide. So the somatic cells, with each round of cell division, they are the length is reducing, 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 reducing. It reaches the Hayflick limit. Now it's like I cannot divide. Now just let me die. Then what happens? Until a critical length of the chromosome, the reduced telomeres are tolerated. So here also you can see the telomeres are getting reduced only. But here the reduced telomeres, they can be tolerated. Once it gets below this, once it get here, it will eventually lead to a crisis phase. Means here also it is getting tolerated, but if it gets below this, it reaches a crisis phase. In crisis phase, what happens? Cells will completely stop dividing unless it gets a second chance in the form of cancer. So telomeres activity has to be turned on. That means here, up till here, it is okay. Here also it is tolerating. Then it comes to the crisis phase. Now in crisis phase, what happens? Cells has to die, or if there is certain kind of mutations that are taking place, some kinds of modifications that has happened, now cells get a second chance, but now they are no longer normal cells. These are now tumor cells. So in the crisis phase, the cells will completely stop dividing unless it gets a second chance in the form of cancer. So telomerase activity has to be turned on. So telomerase activity is turned down in germ cells. It is turned on in stem cells. And when immortalization of the normal cells occurs, that means when your normal cells, they are converted to cancerous cells, at that time, again your telomerase activity is switched on and cells achieve immortalization. Okay, this is the complete gist now. So, and you see, this immortalization is not a very common event. You know, 1 in 10 million chance of an immortal cell overcome barrier for senescence and the crisis M2 phase have the ability to proliferate indefinitely. I mean, after this, the immortal cell is formed, the chance is 1 in 10 million that it will proliferate indefinitely and and then it expresses telomerase, and telomerase is expressed in 80 to 90% of all cancers analyzed and is lacking in most somatic tissues. So this again gives a very new and a very interesting idea of research. That means, so we all study the properties of cancer. We study what are the things that are happening to cancers, this and that. Okay, so Kavita has already answered. Okay, Kavita, so we'll keep your answer on hold. And when we will solve the question, then uh, I'll just keep a note that you have answered the question. Fine. So what I was telling you that we all study so much about cancer. Okay, so Janara has also answered it. Okay, fine. So many different ways in which we study cancer. We have checked the metabolism of cancer, what pathways are cell differentiation and proliferation pathways are activated in cancer. And this is also one more angle in which we can study about cancer, that telomeres length. That means the length of the telomeres in 80 to 90% of the cancers, the length of telomeres is increased. Why? Because telomerase enzyme is activated in, which is again very interesting. So summary, replication problem can be solved with the help of telomeres, and telomeres are 5 to 8 base pair G-rich non-coding repetitive DNA. It adds, and telomerase are the enzymes that add telomeres to the end of the chromosome, and telomere dysfunction can lead to cancer. This is fine. So now, this is just a diagram. if you want to study, you can just see it's nothing much. You can see that up till Hayflick limit it is happening. If the Hayflick limit, that is the M1 phase, is bypassed, then your carcinoma or the tumor formation happens. This is just what we have studied. They have shown in a diagram. Fine. Now, now comes another important, very interesting topic: maintenance and manipulation of telomere length. How telomere length can be maintained or manipulated. This is the last topic, I think, and very easy and very interesting. So, telomere length can be maintained in three ways. What is the first way? Factors that can excel. It is the, see, you are maintaining telomeres and you are manipulating telomeres. So how telomeres can be manipulated? By having factors that accelerate telomere shortening. So if there is telomere shortening, then aging would be increased. Matlab, diseases would be increased. Means aging will come early, diseases would be increased. Factors that slow telomere shortening, something that we can use in anti-aging, and certain telomerase activators that we can add. So, what are the factors that accelerate telomere shortening? So, telomere shortening can be accelerated by one thing that most of the people are suffering today, and that is stress. So, according to one study that has been done, you can see this is the T by S ratio, and for a healthy individual, T by S ratio should be more or less. Tell me, T by S ratio should be more or it should be less? For a healthy cell, it should be,
It should
nobody
be
knows?
more.
I'm sorry, I could not hear you, Jahanara. Can you just type it, please? Okay. Hmm. More. Good. Jahanara, Tarkeshwari, everybody has answered correctly. So, you can see this is high stress and this is low stress. And you can see people suffering from high stress, they have um, reduced T by S ratio in comparison to people who have had low stress. So you see, stress is the cause of so many problems in today's life, and telomere shortening is one such problem. And how you can slow down the telomere shortening? By taking vitamin C. So vitamin C has antioxidants, and these antioxidants are stress busters. So if you take vitamin C, you see people who have had taken vitamin C only in one quartile and who have had vitamin C in all the four quartile, you can see they have greater T by S ratio, or their mean telomere. Okay, their mean telomere length is so much higher than the people who have consumed vitamin C so less. So telomere shortening can be accelerated by stress, and we can slow down the telomere shortening by having vitamin C in our diet. Next thing is, we can also slow down the telomere shortening by having omega-3 fatty acids. You can see this is the T by S ratio, and this is the time duration up to which they have taken the omega-3 fatty acids. So people who have taken omega-3 fatty acids up to fourth quartile, you can see that the length of the telomere is high in them. So how we can reduce the telomere shortening? By having vitamin C in our diet and by having omega-3 fatty acids in our diet. Okay. Now we'll try to solve questions. So these are the questions that are given in your, there are some questions that I have to solve. So I think by now you all know, because many questions are repeated in my and the other TA class also. So that's why I have to solve these questions. Some question might not be given in your theory, but then again you have to solve them. There is no option. So which of the following cause chromosome compaction? snRNA, shRNA, HAT or PRC2? What should be the answer? Think. Try to answer the question. I will switch the screen in 1 minute. Okay. So Kanchana has answered A, Targeshwari has answered C, Madhvi and Janara has answered C. Okay. The correct answer is first, that is PRC2. So basically this PRC2 is I'll just write it here. It is polycom repressive complex 2. Okay. And this is responsible for doing the methylation of the histones. So when this PRC2 is responsible for methylation, so what it does, it maintains the chromosome in a heterochromatin state, because methylation, it reduces or inhibits transcription. So PRC2 is a protein, and the name of the is a actually it's a chromatin regulator, and the full form it's polycom repressive complex 2, and it causes the methylation of the histone, and therefore it causes chromosome compaction because it maintains the chromosome in heterochromatin state. Okay. And then next question is, telomeres can be visualized using fluorescence microscopy, real-time PCR, northern blotting or western blotting. This you should be able to answer because I have shown you one image of telomeres at the very first slide. If you remember this, you can solve the question. Think. Okay. Um, Kavita has answered 8. Sorry, A. Fluorescence microscopy. Yes. Yeah. The correct answer, and okay, I think others have also answered. No, there's only one answer. Fine. So the correct answer is A, that is fluorescence microscopy, and you can see over here. This is the image that I have shown you, where it shows that it is present at the ends, and this is a fluorescence microscopy image. Fine. Next question is, Hayflick number refers to age of a cell, number of times a cell can divide, stage of DNA replication, and cellular senescence. What should be the answer? What does Hayflick number refers to? 1 minute and then I'll switch the screen. Okay. So Kanchana, Madhvi, Bhagya, Kavita, and Janara, everybody has answered B. That means number of times a cell can divide, and it is correct. Next question is, cancer risk is high when telomerase is inactive, telomerase is short, telomerase is active, and P53 is active. Tell me the correct answer. Very easy question. When you think the cancer risk is high? Okay, I'm switching the screens. Let me see. Okay, so Madhvi, Kanchana, and have answered C, and Kavita has answered B. Okay, so the correct answer is C. Why? Because in what happens in cancer cell, there is no shortening of telomere. There is no shortening of telomeres. Cancer cells are dividing indefinitely now. So when they are dividing indefinitely means the telomere is getting renewed each and every time. The telomeres are maintained constant. There is no shortening of the telomere. There is no reduction of the telomeres. So how telomeres cannot be reduced? Only when there is a constant supply of telomeres. In cancer cells, there is a constant supply of telomeres. And what is maintaining this constant supply? Telomerase. That means telomerase is active because cancerous cells are coming out of somatic cells only. Na? See, here we have studied. See, these are somatic cells. Telomerase is inactive. Telomeres are getting reduced, and reduced. Now here some immortalization happens, and now the normal cell gets converted into a cancerous cell. And now it again starts increasing. And here telomerase is active. So in this question, the question was, cancer risk is high when telomerase is active. When telomerase is short, then how cancer risk can be high? The cells will just die after some times. So the correct answer is C. Okay? Is it clear? Kavita, is it clear to you? Okay, good. Next question is, DNA and replication problem is never seen in stem cells. True or false? Okay. The answer is false. Why it is not true? Because problem is there, but that is not that much prominent. See in this graph, unlike germ cells, which is exactly uh, constant, stem cells also maintain approximately a constant behavior only, but over a period of time, ultimately it will also come to senescence. Stem cells cannot replicate indefinitely up to, I mean, they have the property of cell renewal, but up to a certain age, they will also die. But that is very different from somatic cells. I mean, somatic cells are reducing their telomeres at a very fast rate. In an exponential manner, they are decreasing. But you can see reduction is very slight. It's not a very prominent reduction, but we cannot say that there is no reduction. Okay. So the answer would be false here. Okay. Next question is, cellular senescence is not seen in all cell types. True or false? One minute and then I will switch the screen again. Okay. The correct answer is true. Why? Can anybody tell me why it is true and not false?
In cancer cells, it is not seen.
Yes, correct. So, cellular senescence is not seen in all the cells. Of course, in cancerous cells, in germ cells, there is no, I mean, especially in cancerous cell, there is no cellular senescence. Even in germ cells, there is no uh, cellular senescence that is happening because of telomeres. At least for because of telomeres, there is no cellular senescence happening. There can be other factors that contributes to cellular senescence, but telomere is not one of the factors that contributes to cellular senescence in the case of germ cells. Next question now comes this question that I was talking about. So smaller T to S ratio, smaller T by S ratio is a consequence of age, stress, more intake of vitamin C rich foods, all of the above, or both A and B. So I already have two answers,
Both A and B.
both A and B. Okay. So Madhavi, why you think it is all of the above? So if you take more vitamin C rich food, then your T by S ratio would be less or more. Tell me, if you have more vitamin C rich food, that means it is helping you. It is vitamin C is rich in antioxidants, which reduces your stress level, which reduces the oxidative species in your body. So ultimately, these are things that helps in increasing the longevity of the telomeres. Okay, so C cannot be the answer. It is E, both A and B. Is it clear to you, Madhavi? Is this confusion clear to you? Okay, fine. This is just I have included. If you want to go through, you can just go through this end replication problem. I'll just write it here so that if you're going through the slides, at least you know what this is. This is about end replication problem. Just a little more detailed description about end replication problem. So if you want to go through, you can go through. Otherwise, I have already explained this. So what we have understood today, quick revision, very quick revision. What is telomeres? So telomeres are repetitive non-coding DNA sequences which are present at the ends of the uh, chromosomes, and then because of end replication problem, to solve the end replication problems, we have telomeres, and telomeres are synthesized with the help of the enzyme telomerase, and the sequence of the telomeres is TTAGGGG T2AG3 6 hexameric sequence. And then we studied what are telomerase and how does telomerase works. Then we studied that the link of telomerase and aging, and we studied about the Hayflick number, which describes the number of times our cells can divide. And then we see that the telomere length is a mark for aging. If the telomere length is high, aging would not come in such a early time. But if telomere length is less, then you will have early aging. Okay. And then we studied that telomerase affects all the primary, secondary, tertiary, and quaternary structures. And then we studied about this graph, the genetic clock graph, where somatic cells is showing the reduction in telomeres, where germ cells are constant, and here immortalization takes place and it forms tumor cells or cancerous cells. And then we see that you can see that the T by S ratio, the T by S ratio should be high. If it is less, then it might lead to different kinds of diseases. And then we saw that stress can accelerate the telomerase shortening, and telomerase shortening can be slowed down with the help of vitamin C and omega 3 fatty acids. And this is all what we have studied today. So I hope the lecture is clear to you. And we just have one more class, but I will take one more. So we have one more class to cover the course, and last class that I will take your doubts, and I will try to solve some questions. So that would be like a full and final class before your this thing exam. So okay then, all the best, and I hope you were able to understand something from the class.
Yes, ma'am.
Okay then. Chalo, bye. We'll meet next week then.